I'm Robert Bain. I am one of Jeff Kritschmar's PhD students in cognitive neuroscience. This is my first year here, and I'm thinking long term. And in our lab, we have this Carl Sim software that Jeff mentioned in class before, right? And in here, it's a software library for large-scale biologically detailed spiking neural network simulations. We use like leaky, integrate, and fire neurons, and also the Izikevich style neurons models, spiking neuron models we saw from class. Like you can see here on the right, I think these are light years better than the approximations of neurons we use in deep learning. But I think I want to explore making them even more biologically plausible with something like modeling dendrites. Maybe not as complicated as Jeff did during his PhD, but somewhere in between. I think that'd be great. So this is the motivation for this project. I just wanted to include some simple dendrites. Here's an example where dendrites really matter, right? So you see all these are retinal ganglion cells in a mouse's eye. You see they're all asymmetric dendrites pointing downwards, and these are motion selective. In the motion they preferentially detect is a motion going from the soma to the dendrites. Here's the same type of thing, just showing you how the arrows show you where those dendrites point. Here's another, and you see they're motion selective, so they are more responsive to downward motion. And what's really interesting is if you have, instead of this asymmetric dendritic arbor, some of these specific types of ganglion cells are symmetric at the periphery, marked on the right image with a blue star, and they lose their preference to motion. They respond to all types of motion. So that's pretty interesting. Points that the dendrites matter. Old school, going back to the 50s, this is kind of half of a Reichardt detector. So this detects motion. Is theorized to be like in several invertebrate brains, like a beetle or a fly. And so you'd have some feature like this box moving across the image from left to right. And A and B would detect the boxes at different times, but their signals would get to see the motion detecting neuron at the same time and integrate, causing it to fire because there's an axonal propagation delay. If you add kind of another motion detecting neuron and a D neuron, you can actually make a full dual symmetric unit Reichardt detector where you kind of check the sign. When you compare the signal you get from the left and the right here, the sign of the Reichardt detector, whether it's positive or negative, will tell you which way the motion is. And that's kind of what I wanted to kind of implement, something like a simple Reichardt detector. So this is a proposed spiking simple compartmental motion detector model. So this here would, this top right soma with its simple dendrite would be like A, and the lower dendrite below it would be like B. And then this soma would be something kind of like a C neuron or a neuron that wasn't shown before. But essentially what's going to happen here is, let's say we've got this leftward motion of the box. So synapse A would fire. In the top one, synapse A has a stronger efficacy, a stronger weight. So you see this kind of lightning bolt is larger. Time goes on. And then by the time the feature is detected in B, you can see synapse B fires. And already we have this big temporal difference showing up. So that when these finally go all the way down the soma or to the soma, down the dendrites to the soma and integrate, the top one is more likely to fire. Its weight on this third cell is of a different sign. So if the top one's excitatory, the bottom one is inhibitory, and you will be able to detect that influence on the final integrating cell's basal firing rate. And you could imagine, so you could sharpen up the responses, the window of motion, velocity motion that this can detect, by adding something like this, where we know in pyramidal cells and in action potentials that the depolarization, the voltage of the dendrites, can cause more or less signal attenuation. Yeah, so I'll let you think through that on your own. Thank you. If I'm not there today in class, I will check the video for comments, so please let me know what you think. Thank you.